Next, we have Dr. Sathya Venkataramani, a Associate Director at Janssen Biotherapeutics. At Janssen, she leads the biophysics team, focusing on preclinical development of antibody-based drugs, and is responsible for structural characterization techniques and process establishment for selecting manufacturable candidate leads. Today, her talk is focused on how conformation is key to drug discovery and development.
Hello, everyone. It's truly a great pleasure and honor here today to get invited by Nanotemper, highlighting the importance of confirming the conformational stability of biotherapeutics during drug discovery and development phase. In fact, Nano-DSF is one of my personal favorites in our early screening toolkit that we have developed in my team. By the way, I'm leading the biophysics team in Janssen Biotherapeutics, which is located in Spring House, Philadelphia. Let's cut to the chase. Why conformational stability determination is very important in the early screening phase. So let's take the R&D productivity scale, right? The topic of R&D productivity has been discussed over and over again for more than 2 to 3 decades by now. And undoubtedly, monoclonal antibodies have dominated with such a medical breakthrough in this industry so far, with a steady growth in the past 10 years. Commercial pipeline, in fact, is very robust with more than 60 monoclonal antibodies in late-stage clinical trials. However, 75% of the drugs that enter the phase 1 clinical trials are destined to fail, with only 25% successfully gets approved for treatments, as you can see here in this graph. There could be major reasons like many reasons like wrong mechanism of action, wrong target, and wrong patient population. But one of the major reasons that I think is also for this decline is poor intrinsic properties, which truly takes a big stab on getting approval. So what actually makes a very successful antibody drug? During the generation of antibodies, for successful progress from discovery to becoming an approved drug, there are many intrinsic properties that are critical. And we all know those major characteristics like affinity, function, safety, manufacturability, serum half-life, etc. One of the very famous papers that I think that was published in 2016 that rocked the scientific world is from Adimab, and we all are aware of that. What the research group done from Adimab is they took this 137 clinical stage antibodies and subjected those antibodies to 12 different biophysical assays, which are commonly used for developability assessment. And comprehensive analysis of this set of antibodies has shown significant developability risks. However, such number of red flags decreased as the monoclonal antibodies advanced to the approval stage, which clearly demonstrate here that selecting a molecule with better biophysical profile is very critical for the success. It is well known that the drug quality is an immunologically dominant factor to maintain its stability, efficacy, and safety, right? And the key attributes that to ensure that selected lead maintain its biophysical and biochemical profile throughout the drug discovery and development process include thermal and chemical stability, aggregation potential, PI of the lead, and also charge heterogeneity. And it's very critical to address these intrinsic properties early on to select a lead molecule that won't have manufacturing headaches later on. So here is a typical screening funnel from discovery to development for candidate selection and characterization. Of course, it takes anywhere between 1 to 3 years, even beyond that in some companies, depending on the lead molecule, to be discovered and to be progressed to the development phase. Topmost quality attributes clearly include binding affinity and specificity to the target, including some of the in vitro functional characterization. Then right away, what the project team does, it pushes the molecules that meet these minimum criteria for a complete manufacturability assessment. Then again, depending on the resources that we have, the novelty of the target, the timeline, the number of lead molecules that we have to characterize, the team split the work into high throughput funnel as well as later on into in-depth analysis. Overall, this process actually takes a couple of months to clearly make decisions on selecting the lead molecules without liabilities. Here, under HTP workflow, I have placed conformational stability assessment, which comes from the parameters TM and TAG by NanoDSF as the topmost assay. And you will see why in the upcoming slides when I describe some of the case studies. So with that, let's move into the case study 1 to show Fab stabilization is very critical. It's truly an interesting story about a bispecific antibody. Here is the overview of the high-level screening funnel for the antibody that was selected against target 1. Of course, immunization followed by initial screening led to the identification of a lead molecule that met the criteria for in vitro pharmacology, but not for biophysics. And when we took a closer look at the sequence of the lead, it showed some rare somatic hypermutations. So hence we further optimized this candidate, which retained function, but also significantly improved its intrinsic properties. Particularly, thermal stability played here a major role in selecting the lead. So why stabilizing Fab is very critical? Of course, it's well known, right? And here I'm highlighting only 3 papers, but there are much more papers to show increasing Fab TM is overall a better approach to improve pharmaceutical storage stability. In one of the recent studies, the group has shown that the monoclonal antibodies where they had the Fabs that unfolded along with the CH2 domain showed lower stability at accelerated stress conditions, while the MABs where the Fab melted independently of the CH2 domain with a higher TM had exhibited lower monomer loss at 40C storage, which predicts the shelf life. And there are also a couple of papers showing a very positive correlation between thermal stability and serum half-life. What I'm showing here is a data set of engineered variants and also the TM and TAG measured for those purified variants. And the difference in the TM and TAG of the variants compared to the wild type is plotted here. And the different color represents the different engineered variants of the wild type. Here, the EpiVax score is plotted for the heavy chain and light chain of these variants. EpiVax company, I'm sure most of you know, it uses its proprietary state-of-the-art immunoinformatics tools and in vitro techniques to predict the potential immunogenicity of proteins. EpiVax screens for immunogenicity and it employs a proprietary scale that compares the protein to antibodies which are already in clinical use. So based on their scale, the light chain as well as the heavy chain scores are plotted here. Taking into account both EpiVax score and TMTAG, we selected PCA62, which is shown here in green, that not only exhibited ideal EpiVax score, but also enhanced TM and TAG almost by 12 to 15 degrees Celsius, as measured by NanoDSF compared to the wild type. Here, what I am showing is just confirming that conformational stability done by DSC. P5 is the wild type, and P2, which is shown here in orange, is the optimized variant. Clearly, you can see the shift in the TM of the Fab from 60 degrees to 75 degrees Celsius, which was already clearly measured by NanoDSF before, also shown by the enthalpy of unfolding here. Okay. Of course, a high thermal stability is commonly beneficial for therapeutic proteins to maintain the active native state at physiological temperatures. But there is one more important factor that needs to be considered, which is free energy of unfolding of an antibody, even at lower temperatures. Because a subtle change in conformation can eventually lead to aggregation, loss of activity, and high immunogenicity risks. So in my mind, measuring inherent stability of an antibody, even at lower temperatures, is one of the effective ways to understand and select a therapeutically viable candidate that can truly withstand all stresses during manufacturing, storage, and shipment. And one way to measure that is through the chemical denaturation, which we again performed using our nano-DSF instrument here. So conformational stability is defined by Gibbs free energy of unfolding between the native and denatured state. The blue curve shows it for the wild type, and the P2 is the optimized variant, as I have explained before. You can clearly see PCA, this P5 is exhibiting two-state unfolding, while the P2 is showing a three-state unfolding. And the initial inherent stability, as shown by the Gibbs free energy of unfolding, is 24 kilojoules per mole for the wild type, while it is 63, almost doubled for the P2 variant. So intrinsic stability of the optimized variant has also significantly increased, as shown by the chemical denaturation experiment conducted by nano-DSF. Here is yet another validation for the aggregation potential of these two candidates. What we did, we took the P2 and P5, the wild type and the optimized variant, just at 1 mg per ml, kept it in simple PBS buffer, and monitored the integrity of this molecule by analytical SCC over time at 4C as well as 40C for a month. And the delta difference in the aggregate between the starting sample and the incubated sample is plotted here. It's very clear that the sequence optimized P2 has also very low aggregation potential for a month at 40 degrees Celsius, predicting good shelf life. So overall, in this project, our initial screening of the engineered variants by nano-DSF clearly showed the selection of the best variant that exhibited ideal biophysical properties with respect to intrinsic stability as well as manufacturing. And currently, this molecule is in clinical trials. With this, let's go to case study 2 to highlight that heterogeneity or product purity also affects the TM profile. In fact, it's also one of the very interesting project for us. Here, what we have done is we considered SCFV. Obviously, it's a bispecific antibody. And this SCFV was constructed in 2 different orientations, heavy to light and light to heavy, for constructing the bispecific antibodies. And so let's start calling this as SCFVX and SCFVY. The sequence is the same, just the orientation is different, okay? And these 2 were subjected to biophysical characterization to determine the quality. And the first thing, as I have mentioned before, we always do is the nano-DSF for TM, TAG, as well as to understand the profile, the conformational stability of these molecules. So purified SCFVs were subjected for the nano-DSF experiment, and the table here gives you the TM values as well as the TAG calculated from this profile. What is very interesting here to note is the thermal unfolding profile shows that the molecule Y already shows an early unfolding event close to 50 degrees Celsius. And the overall thermal profile has shifted towards the lower temperatures compared to X, right? So this itself already indicated to us that there could be some heterogeneity or some impurities associated with these molecules. And hence we took these 2 molecules and subjected to various biophysical characterization to clearly understand what is causing it. And this table gives you the purity assessment done by analytical ultracentrifugation, self-association by AC sense, non-specific binding, as well as serum stability for both X and Y. It's very clear from the assessment. It shows that the molecule Y already had like 8% of fragment, which got co-purified along with this molecule. And the thermal stability, as I have shown, already had a minor unfolding at 50 degrees Celsius. And also, this molecule, when incubated in human serum, within 2 days, started to show 16% of aggregation propensity. So clearly, SCFVY had poor intrinsic properties, while SCFVX meets our criteria. But because of the novelty of the target, what we did was we moved forward both molecules for in vivo efficacy study. And however, SCFVX was found to be efficacious compared to Y. So this is yet another example to show that a true correlation exists between in vitro assessment and in vivo behavior, highlighting that our team is really equipped with the right toolkit to evaluate the intrinsic properties of the leads very early on. With this, I'm going to move to the final case study 3, where again, it's also another very, very interesting project to show that the conformational changes truly reflect the physiological behavior. As I mentioned before many times, nano-DSF is one of my personal favorites biophysical characterization tool, and its intrinsic fluorescence is truly most powerful method for detecting conformational changes and to determine the conformational as well as colloidal stability. Okay. So for this lead, we focused on early physiological behavior assessment because it's really critical for selecting the right lead molecule. And the key assays here included serum-based physiological stability determination. And the techniques that we used include SPR, nano-DSF, and mass spec, and also including cell binding. This whole assessment took only 2 weeks with few micrograms of material requirement. But really, you will see from the data that it was very critical for us to filter out the molecules with undesirable biophysical properties. Here, what you're seeing here is the nano-DSF profile of the lead molecule that was incubated in PBS at different temperature 4C as well as 37C over time. And aliquots were taken and they were analyzed for conformational and colloidal stability. The first derivative plot is shown here at the bottom panel and the ratio is shown at the top. Arrow indicates that Fab melting, which clearly has shown some conformational changes associated with this particular domain over time when it was simply kept in PBS at 37C. So that's a concern for us when this molecule is clearly showing some conformational changes in the Fab. That's why we did other characterization to truly understand its binding to the target and how it affects it. So we moved forward. We took this lead. We incubated it in serum and again in PBS at 37C. Time-dependent aliquots were taken to characterize the binding of this molecule to the antigen by a simple SPR experiment. And in this, what you're seeing here is the percent relative activity is plotted against the aliquots that were incubated in PBS at 2 different concentrations or in serum, again at 2 different concentrations over time. Initially, the percent relative activity calculated to be 100%, and relatively, you can clearly see a decline in percent active species over time in both buffer and serum for this molecule. That's again another concern for us. So hence we moved forward and asked the project team to also do the cell binding. Cell binding was assessed for this molecule using 2 different target cell lines that had different expression profile. What you're seeing here is different colors, blue, red, and green that represent the lead incubated in PBS at different temperatures, while the purple shows the binding of the sample in 50% human serum. The table gives you the calculated EC50 and EC90 values for 2 different cell lines for different aliquots. From the EC50 or the EC90 values, there is a significant drop in potency of this lead molecule binding to the target. And obviously, it is associated with the conformational changes in the Fab domain that we have already determined by the nano-DSF tool. And very important question for the project team is like why it is happening and what is causing it in the Fab. Obviously, mass spectrometry is one of the best methods to characterize that. So we identified from the sequence there is a very high risk DG in the CDRs of the lead molecule. And the reason for such a conformational change can be clearly explained by the presence of this DG, which was undergoing a significant isomerization over time. What you're seeing here is the ion traces that are plotted to show that molecule undergoes almost like 48% of DG modification that caused a huge shift in the DSF profile that caused also loss in percent activity by SPR as well as drop in the potency by cell binding. So what we did was immediately de-risk the molecule by engineering and did a quick SPR as well as DSF of the molecule, both at pH 5 stress as well as physiological stress, to show that this DG de-risked molecule retained binding to the target as well as exhibited stable conformation, as you can see by the DSF trace, nano DSF trace here. Okay. So this molecule is also now part of the bispecific antibody moving into development very quickly. In summary, with rapid progression towards drug discovery and development, industries are constantly looking for shrinking timelines, using minimal resources and achieving fastest timeline towards taking high quality drugs to the clinic, right? Hence, having early de-risking toolkit is an absolute must for industries to filter out the bad ones and to select only the manufacturable molecules. In the efficient, practical HTP workflow that we have created in our team, we always place conformational stability as one of the most valuable assessment done very early on for lead selection that requires only few micrograms and can be done within few days, including data analysis. With this, I'm coming to the most important slide here. In our biophysics team, we strongly believe in not wasting our pure thoughts on any impure proteins. We live by our mantra, which is play to win, don't play not to lose. So I sincerely thank Janssen Biotherapeutics for providing the support and resources, and also believing in what we believe in our team. I'm also extending my thanks to our TA partners for wonderful collaboration, and finally to Nanotemper, not just for inviting me here to talk about the case studies, but also that team is constantly working to innovatively discover and develop great instruments and tools that enable biotech and pharmaceutical companies to select the first time molecule in discovery research. Thank you once again for your patience and attention.